Sorry, let's continue. So the first structure feature is the inner membrane is impermeable to hydrogen ions to create the electrochemical proton gradient. Don't just stop there, elaborate with effort, okay? For chemiosmosis to occur. Second structure feature, Josh, inner membrane, just like your thylakoid membrane.
Like yes.
Highly.
And then like there are like ATP synthase.
So highly folded. Okay, so why must be highly folded, dude? To increase
surface area for the environment of the ATP synthase.
only and
the
electron carriers. Okay, so increase surface area, okay, for increased abatement. And ATP synthesis. Okay, number 3. Number 3, we have this keyword. Please know this keyword from now onwards. Compartmentalization. Jane, when you have compartmentalization, what is the purpose of the compartmentalization? To provide what? Optimal. Conditions.
Conditions for the different processes.
Yeah, like what? What are the two?
Cycle
And
oxidative phosphorylated.
also in the liquid. Yes, okay. So to provide optimal conditions
No, like
in the inner membrane. Wait, let me write here. To provide optimal conditions for enzymes. You know why? One main feature we already established, like what we learned in photosynthesis last week, is the difference in concentration of hydrogen ions, right? We also have difference in concentration of hydrogen ions in the mitochondria. Josh, can you tell me the hydrogen ions is higher where? Is it in the matrix or in the intermembrane space? Since you should know this, called the matrix.
it's like higher in the matrix.
Higher in the matrix, 你的头啊. H plus ions is high here. Oi, you know where is your ATP synthase complex facing or not? I give you a tip. How you remember is, I want you to remember where the ATP synthase complex is facing. So where is the ATP synthase complex facing in the photosynthesis first? For the chloroplast. Facing where first? You just did this. When I draw, every time I draw inwards or outwards.
Outwards.
So it's facing outwards, right? Into the... Yeah, so do you think this is the opposite?
Yeah.
Yes, so the ATP synthase complex is facing where?
Like inwards.
Inwards, right? Okay, so you think about this, why I asked you to learn your ATP synthase complex where it faces is because you know where it faces, it shows you the diffusion direction of H plus ions. Do you agree? So diffusion always occurs from region of higher concentration to region of lower concentration. So that's why we know H plus ions is diffusing in this direction. Understand? Clear? That means pumping pump where? In which direction?
Like into the intermembrane space.
Correct. You understand? So how you remember from now onwards, class? Just know where the direction in which ATP synthase is facing. Okay? Now, anyway, based on this, do you agree H plus concentration is already high here and low here? So it means that your enzymes, right, in your matrix must be optimal, right, for your this high pH, right? pH is going to be one factor already, what? So that's why compartmentalization is very important. Clear? Clear? Alright, so again, what's the three? First one?
Create the electrochemical proton
How?
by
Being
impermeable to H+.
Second one is what, Josh?
By refolded
Uh-huh.
for
Increase
surface area for
Uh-huh.
the embedment of
more.
electron carriers and ATP synthase.
Then next one, keyword, start with C.
Compartmentalization.
Why?
By optimal conditions for different processes.
Yeah, which
In the
is
matrix.
understand in a matrix. Alright, next one. I forgot to talk about this since we're already talking about chemiosmosis, right? And you know what? I'll do when I go through this chapter. Okay, so next one. Whenever you do this aerobic respiration, alright, today we'll just focus on aerobic respiration. We learned that this respiration always comprised of glycolysis, okay, your link reaction, your Krebs cycle, and then finally my oxidative phosphorylation. And all of them actually involve, okay, the electron carriers NAD plus and FAD. However, what I do not give, and they have asked before in one prelim paper, I can't remember which, NY. Okay, they asked what is the significance of NAD plus. Okay, find some space to write the significance of NAD plus, which is also the same thing as your significance of FAD. Alright, both same. Ready? So significance of NAD plus, you can write, first bullet point is NAD plus receives electrons from substrates in, I repeat, receives electrons from substrates in glycolysis, link reaction and Krebs cycle, receives electrons from substrates in glycolysis, link reaction, and Krebs cycle, and is reduced to, either you write reduced NAD or NADH, that carries high energy electrons to the ETC, that carries high energy electrons to the ETC, at the cristae, underline for me the keywords first, receives electrons, reduced to NADH, carries electrons to ETC. Second bullet point, you're right, where energy release during transfer of electrons down ETC, where energy release during transfer of electrons down ETC is coupled to
The electron when energy release
during transfer of electrons down ETC is coupled to formation of proton gradient, will be harnessed by ATP synthase, to power chemiosmosis. Okay? Anyway, now we already learned all this four. Today I'll be focusing on glycolysis. If I got time, then I'll move on to others. I need to point out, in exam, you are not expected to know every single step of glycolysis, but everything that I'm going to write on the board, you are expected to know in syllabus, or at least good to know. To me, know all. So, first thing on glycolysis, this is actually in your notes on the 4th column of the first page of your notes, okay? Jiaxuan for you will be your second page of your notes, second column, okay? And then this will also be located on the page, second page of your notes as well. First step in glycolysis is, your glucose is always the first step. Your glucose is first converted to this thing called glucose 6-phosphate. I do want my students to know the name of the reaction, which is obviously called phosphorylation. Please know this step, it is necessary, explain why later, okay? And I also want my students to know it's catalyzed by an enzyme called hexokinase. Why? Because hexokinase and phosphofructokinase, okay, there are two enzymes they usually like to test for exams. So at least knowing what the hexokinase do is useful for you to know. This is diagram must know. This glucose, notice how there's a phosphate added, right? Where does the phosphate come from? It actually comes from ATP. So they kind of transfer the ATP from phosphate from ATP, okay, to glucose to become your glucose 6-phosphate. For two reasons. Number one is obviously to increase the energy level, okay, of your glucose. But the second reason, can you make a guess, Jane? Why is it necessary to actually convert glucose to glucose 6-phosphate? No. I already said already, not only to increase the energy level, but the second is actually to maintain a low concentration of glucose within the cell. Why? You always want a continual diffusion of glucose into the cell for respiration to occur. Understand? But the next thing is, you are not expected in syllabus to know the next step, but you have another round of phosphorylation. The next round of phosphorylation is going to form fructose 1,6-bisphosphate. Notice how in your notes I have this enzyme called PFK, stands for phosphofructokinase. This is another round of phosphorylation. I want you to make an asterisk of phosphofructokinase for me, this PFK enzyme, and write at the side that this serves as an allosteric enzyme.
Wait, it's to maintain low glucose concentration in the
Cell, cytoplasm, cytosol. Because you should know, glycolysis, notice how I deliberately write outside, it takes place in the cytosol. Because in the cytosol is where your glycolytic enzymes are located. That means like all this, hexokinase, PFK. Okay?
Why do you always want to you expire?
Yeah, I need to expire. No, I die. Okay, so please tell me at the side, right, for me, this PFK is an allosteric enzyme. Now it's allosteric to what? Allosteric to ATP. Okay, we always call that this step is the control pathway of glycolysis because not only your PFK is in low concentration, it's allosteric to ATP, meaning at high ATP concentration, activity of PFK is low because you already produce sufficient amounts of ATP. And if you have low amounts of ATP, your PFK activity is high. Okay, so it's allosteric. As I said already, this is actually phosphorylation. So you should know already glucose becomes fructose, that means this step must be isomerism. Okay, but another round of phosphorylation where a phosphate group is transferred from ATP to the fructose to become fructose 1,6-bisphosphate. Okay, now you notice how they will undergo splitting. It's not in your syllabus too yet. You split to become two molecules like this, but eventually they will actually reform to form G3P. So essentially, how many G3P will I form? 2. Understand? So I want to focus on this first. So this step is what we need to know. This step is called dehydrogenation. Dehydrogenation is a step not expected for you to know the enzyme for this case. But dehydrogenation is a step where your NAD plus will help to further oxidize, okay, the glucose. So I will become 1,3-diphospho glycerine.
Between the fructose that 1,2,G3B is nothing.
Oh, splitting, lysis. I say it's not in your syllabus to know. But everything I write aside from the name of the reaction is good to know. Okay? Now, where was I? Oh yeah. So, again, like I said already, you will actually form, okay, your this dehydrogenation, undergo dehydrogenation. Okay, so your NAD plus acts as a coenzyme here. That's why you can form your this NADH. But my question is, Josh, can you tell me how many G3P do I form ultimately? Oh, two. Two what? So how many NADH do you think you ultimately synthesize? Two. Understand? Okay? Because I said already, one important thing, if you cannot remember the step, but one thing you really must know for each step is always the products produced per glucose. That's why do you see in your notes that I have in box in blue? Everything that I box in blue is die must know. I already boxed in blue and even highlighted for you in the notes. So the products produced per glucose. For glycolysis, you can see the first one I have is actually 2 net ATP. So let's see, I understand now why 2 net ATP, but I say that I form 2 NADH as well. This is the reason why. But let's continue first. You're not expected to know the product form, but ultimately, you know I will form pyruvate, right? Since I form 2 G3P, every 2 G3P becomes 2,1,3 diphosphoglycerate. Each of your this 1,3 diphosphoglycerate will actually undergo 2 rounds of SLP. I lazy to write, but SLP actually stands for substrate level phosphorylation. Substrate level phosphorylation means what? It means you transfer the phosphate group from the substrate to ADP. I repeat substrate level phosphorylation is you transfer the phosphate group from the substrate to ADP. Why? Do you see di means 2? So you have 2 phosphate in each of this, right? So one of the phosphate transferred to here, you will become ATP. So again, do you agree they have another phosphate group in each compound? So they transfer the next phosphate group, okay, to your ADP to become ATP. But how many of your this compound do I have in total? 2. So how many how many ATP can I synthesize each time? 2. So now let's see. Josh, what's the total number of ATP synthesized so far? 4. Yes, 4, right? But we use up 2 here initially. That's why the keyword must be 2 net gain. That's why you see in your notes. If you never write net gain of ATP, I can guarantee you will never get credit. You understand? So be very careful, must always write net. Alright, last one. Do you see how I must always synthesize? 2, pyruvate. Why? Because every one of this molecule will become pyruvate. Since 2 molecules are being formed, 2 G3P was formed, 2 pyruvate molecules must be formed as well. Okay? So one thing I just want to emphasize here, today I know I just stopped halfway, but today I'll just introduce for you this mitochondria. One thing important they like to test the enzyme is your inner membrane of mitochondria, the features and the function of the features. Please know the terminology here that I write in both in black and blue. Second thing is glycolysis. Every single step in glycolysis, you're not expected to know all steps. But everything that I have in your, on the board here, please know them well. Understand? So I'll stop here for today. Make sure you know the products produced per glucose for glycolysis at least. I'll continue this on next week when I see you. Huh?
How many is it if we test us like that? To outline the whole process. Then if we
outline whole process, sometimes it's your this paper too.
So we need to write all those.
Yeah, I will go through this next week. So there will also be a shortened one. This one, right, is for your content must know. But the shorter one, right, usually is worth 3 marks. I'll tell you what you write next week, so we don't confuse. Okay? So good to know now for content. But to save time I prepared the MCQ because by right your school already went through this topic, okay, for MCQ. Alright. Please complete the MCQ by next week. I'll go through once I finish this chapter, so it saves time that I can quickly go through the answers. Alright, and with that, I end off here.